Hello, everyone, and welcome. In the current COVID-19 crisis, the FDA has taken a very aggressive approach by quickly authorizing many medical devices and diagnostic tests for emergency use. I've been keeping a close eye on that, and I have produced several video updates. You can find them on my YouTube channel or by following me on LinkedIn. Lately, I have been focused more on testing because that's most relevant to our conversation now as we begin to think about reopening our economy. FDA has authorized many different types of tests, but it wasn't until last week that the first antigen-based test got authorized. This is a very significant development because it can open up rapid deployment of point-of-care testing, which can be very fast and it can produce quick results. So in this video, I will give you an overview of the test that was just authorized, a little bit more background about other different types of tests, and then finally discuss a few key points that you should keep in mind while trying to figure out how well these tests are performing in the real world. So let's look into that. There are three different types of tests for the SARS coronavirus, and here is a particle structure for the virus that we will refer to in this slide. The first category is RT-PCR test, and most of the FDA authorized tests use this technique. They detect the viral RNA, which is the single strand genetic material inside the particle. They are very sensitive and specific because they are done in sophisticated laboratories with advanced equipment and trained people. But as a result, they tend to be slow, and there are very few options available for rapid point-of-care testing. One exception is the Abbott ID Now test, which can produce results within minutes. The second category is antigen tests, and they go after the viral proteins. So you have the S proteins, the spike protein here, the membrane proteins, which are the M proteins, and the nucleocapsid protein, which encapsulates the viral material. It's called the N protein. These tests tend to be medium sensitivity and specificity. So there's a concern, but the benefit is very rapid availability of point-of-care testing. One example that we're going to talk about is the Quidel Sofia 2 test. The final category is antibody testing, which detects the presence of antibodies in our system in response to the infection. So you have IgM antibodies, IgG antibodies. These tests also tend to be of medium sensitivity and specificity, but again, you can get very rapid results in point-of-care settings. There are also more accurate antibody tests which have been authorized by the FDA, and they are performed in sophisticated laboratories using highly automated equipment. So both options are available. One example is the Celex Q rapid test that was the first antibody test authorized by the FDA. And again, these are only examples. We are not endorsing any of these tests. We are using them only for illustration purposes. So let's look into the antigen test that got authorized. This is the Sofia 2 antigen test. EUA date was May 8, and it uses a lateral flow fluorescent immunoassay technique. It uses the Sofia 2 instrument. Samples are nasal swabs or nasopharyngeal swabs, very similar to the RT-PCR test. It goes after the nucleocapsid protein of the SARS virus. It is authorized for high to moderate complexity CLIA certified lab or in point-of-care settings like a doctor's office if a waiver has been obtained. And it takes about 15 minutes because the reagents have to do their chemistry to get to the protein, and also the measurement has to take place over some time. But it's still, compared to the RT-PCR test, is very rapid. Look at the test performance at a high level. Sensitivity, which is also the positive agreement, is at 80%, but the confidence interval is broad, 68 to 89%. Specificity is the negative agreement, and it's at 100%. Confidence interval is very tight. What I like to look at are positive predictive values and negative predictive values, and I've talked about those metrics in another video. They are dependent on the prevalence. Right now, we don't know what the prevalence is, so FDA has been looking at this data at 5% prevalence, so I like to look at the data at the same level. The positive predictive value is at 100%. Negative predictive value is at a 99%. But notice the confidence intervals right here. So I did a simulation of 1000 random tests to figure out what the range of NPV and PPV would be. My numbers are 71% positive PPV at an average level with a range of 49 to 100%. Negative predictive value, I came up in an average of 99% in a much tighter interval of 98 to 99%. So let's talk about what it all means. So as I've discussed before in my video, positive predictive value is the probability that when the test is positive, the person in this case actually has the viral antigens, the viral proteins. It is different from the positive agreement that you hear about in terms of the sensitivity of the test. Negative predictive value is when the test is negative, the person does not have antigens. It's a probability estimate, level of uncertainty. Very different from specificity and sensitivity. So it gives us a sense of how likely we are going to be wrong when the test result comes out as either positive or negative. This determination has to be done carefully and the interpretation has to be done carefully because we can make a wrong decision about the treatment or the care for that particular patient. And that's why the role of the doctor is very, very important. You might hear a lot of stories in the media about one test not being as good as another because they're just comparing the sensitivity and specificity numbers. That is not correct. In my opinion, all of these tests have a purpose. They have a benefit-risk balance. And in this particular case, it is a good test. It's available for rapid deployment at point of care, very quick results, but interpretation has to be made by the doctor and it's clearly spelled out in the instructions for use. The doctor decides based upon the patient's conditions, their history, and their symptoms. And they may choose to do a confirmation using RT-PCR test. It's up to them. So I would not be too concerned about a lot of media stories about tests and their accuracies. It is still evolving and more options we have, the better it is in my opinion. If it were my family member, I would be very happy to see that a rapid point of care test is available now. And I will ask the doctor what it means instead of me trying to figure it out. So I recommend that for all of us. There's a lot going on. As you can see, many tests have been authorized. Hundreds of tests are coming out and FDA is working very, very closely. Hopefully, we will have in-home test capabilities soon that can be very quick, very robust, very reliable. So we look forward to that. The best way to keep in touch is to subscribe to my YouTube channel because I follow this very, very closely. And here's how you can do that. You can find my YouTube channel and subscribe to it, or you can follow me on LinkedIn because every time I post something, I give you a link. So hopefully this is useful to you. And if you have any questions and comments, please reach out to me because I will look into that and produce another video update to provide my perspective. I hope all of you are keeping safe and I thank you for your interest and attention.